For a very long time, scientists believed a simple but stark fact. We are born with all the brain cells we will ever have. This idea suggested that our brains were fixed and unchangeable after childhood. If you lost brain cells, they were gone forever. But over the last few decades, we have discovered something truly remarkable. Our brains can continue to create new neurons throughout our lives. This process is called adult neurogenesis. It is a sign of the brain's amazing ability to adapt and change, a quality known as plasticity. This discovery has completely changed how we think about brain health, aging, and recovery from injury. This exciting process of new neuron birth happens in a special part of the brain. It is a seahorse-shaped structure deep inside our temporal lobes called the hippocampus. The hippocampus is incredibly important. It acts as the brain's main hub for learning and memory. When you learn a new skill, remember a special event, or even find your way around a new city, your hippocampus is hard at work. The creation of new neurons in this specific area suggests that our ability to learn and form new memories is not static. It can be refreshed and renewed, giving our brains a kind of fountain of youth. The new cells that are born in the hippocampus are called immature dentate granule cells, or IMGCs for short. Think of them as baby neurons. They are fresh, full of potential, and ready to be shaped by our experiences. These IMGCs are found in a part of the hippocampus called the dentate gyrus. This area is like a nursery for new brain cells. These young cells are very flexible and excitable. They are more likely to connect with other neurons and form new circuits. This makes them critical for helping us learn new things and adapt to a changing world. Understanding these cells is the key to unlocking the brain's potential. Why are these IMGCs so important? Because they represent hope. They show us that the brain has a built-in mechanism for repair and renewal. For people who have suffered brain injuries or are living with neurological conditions, this is a powerful idea. If we can understand how these baby neurons grow and connect, we might be able to encourage the brain to heal itself. This could lead to new treatments for memory loss, depression, and other disorders that affect the hippocampus. The study of these tiny, immature cells holds enormous promise for the future of medicine and our understanding of what makes the human brain so special. To truly understand what makes our brains unique, scientists often look to the animal kingdom for clues. A recent groundbreaking study did just that. Researchers compared the development of these immature dentate granule cells, or IMGCs, in humans with those in other mammals. The study included rhesus monkeys, pigs, and mice. These animals are often used in research because they share many biological similarities with us. The scientists wanted to see if the process of creating new neurons was the same across these different species. What they found was both expected and surprising. The basic biological steps were similar, but the details were very different. The researchers used incredible new technology to peer inside individual cells. One of these tools is called single-cell RNA sequencing, or sRNA-seq. This technology allows scientists to read the genetic instructions, or RNA, inside a single cell. It gives them a detailed snapshot of what that cell is doing at a specific moment. However, finding these rare immature cells, especially in primate brains, has always been a major challenge. To solve this problem, the team developed a powerful machine learning algorithm. This smart computer program was trained to accurately identify the IMGCs from among thousands of other cells, a task that was nearly impossible before. With this powerful combination of technologies, the scientists made a key discovery. They found that the genetic activity in the young IMGCs varied much more between species than in their fully grown mature counterparts. The mature cells in humans, monkeys, pigs, and mice were all quite similar. But the baby neurons were a different story. The human IMGCs showed a unique and rich pattern of gene expression that was not seen in the other animals. This suggests that the journey from a baby neuron to a mature one follows a special path in humans. It is a path that may be responsible for our advanced cognitive abilities. This discovery tells us something profound. While we share a common blueprint for building a brain with other mammals, evolution has added some unique features to the human version. The differences are most obvious during this critical stage of neuronal development. It is as if the human brain keeps its new neurons in a longer, more complex training program. This extended period of immaturity might allow our experiences and environment to have a greater impact on shaping our brain circuits. This could be one of the secrets behind our species' remarkable capacity for learning, language, and complex thought. As scientists delved deeper into the genetic differences between species, they uncovered something specific and fascinating. They identified several families of genes that were much more active in human immature neurons compared to those in monkeys, pigs, and mice. Among these, one gene family stood out clearly. It is called the ATP6 gene family. This might sound technical, but its job is quite important. These genes provide the instructions for making proteins that act like tiny pumps. They help transport protons across cell membranes, a process that is vital for managing energy and communication within the cell. This family of genes was uniquely enriched in the human IMGCs. This finding was a huge clue. The fact that the ATP6 gene family was so active in human baby neurons suggested it must be playing a special role. To test this idea, the researchers turned to another advanced tool, a human stem cell model. They grew human brain cells in a lab dish to create a model of the hippocampus. This allowed them to study the development of human neurons up close. In this model, they could control the activity of the ATP6 genes. They wanted to see what would happen to the young neurons if these special pumps were not working correctly. The results of their experiment were clear and direct. When the scientists interfered with the function of the ATP6 proteins, it had a dramatic effect on the developing neurons. The young cells struggled to grow their branches, which are called neurites. These branches are essential for forming connections or synapses with other brain cells. The experiment showed that the ATP6 gene family is critical for helping human IMGCs reach out and build the complex networks needed for brain function. This provides a direct link between a unique human genetic feature and a fundamental process in brain development, helping to explain our brain's incredible networking power. So what does this all mean for us? This research suggests that the unique activity of genes like ATP6 may be a key reason why the human brain has such amazing plasticity. This enhanced ability of our new neurons to grow and form connections could be the foundation for our superior learning and memory capabilities. It helps explain how we can continue to learn and adapt throughout our lives, from mastering a new language to creating complex art. The ATP6 secret is not just a scientific curiosity, it is a piece of the puzzle that explains what makes the human mind so powerful and adaptable. The discovery of these unique human gene patterns is more than just an interesting fact. It has profound implications for our health and for the future of medicine. Our brain's plasticity, its ability to change and form new connections, is central to everything we do. It underpins how we learn, how we remember our past, and how we adapt to new challenges. The finding that human-specific genes like the ATP6 family help regulate this process opens up exciting new possibilities. It suggests that these immature neurons could be a target for future therapies aimed at boosting brain function and promoting recovery from injury. Imagine a future where we could develop treatments to encourage the growth of these new neurons. This could revolutionize how we approach conditions like Alzheimer's disease, where memory is progressively lost. It could also offer new hope for people who have suffered a stroke or a traumatic brain injury. By understanding the specific genetic on switches that control human neurogenesis, we might be able to create medicines that help the brain heal itself from the inside out. This research moves us one step closer to that goal, highlighting the potential of regenerative medicine to repair our most complex organ. This study also sends a very important message to the scientific community. For many years, research has relied heavily on animal models, especially mice, to understand human diseases. While these models are useful, this work clearly shows their limitations. We cannot simply assume that what happens in a mouse brain is the same as what happens in a human brain. The unique genetic patterns found in human IMGCs would have been completely missed if scientists had not looked at human cells directly. This highlights the critical need to use human-based models, such as stem cell cultures and donated surgical tissue, to truly understand human biology. Looking ahead, the journey of discovery is far from over. Scientists now plan to use even more powerful technologies to explore these cells in greater detail. They want to look at genes that are expressed at lower levels and analyze how different cells communicate with each other. They also aim to study how neurogenesis might differ between sexes or in various disease states. By combining these advanced techniques and focusing on human-specific biology, we can continue to unravel the mysteries of the brain. Each new discovery brings us closer to understanding ourselves and finding new ways to protect our most precious asset, our mind.